Chapter 15, Trematoda, Form and Function. Part 3, the Tegument. Now, when we were talking the integument of the protists, we were referring to this sugar protein shell around the single cell. It had some flex to it and whatnot, but it was there as a shell to add for protection. But here, as we're talking the flukes, the tegument is not this proteinaceous sugar-based shell, because remember, we're talking about a multicellular, multi-tissue, multi-organ system organism. So its tegument is actually going to be more closely related to what we have. A complex system of tissues, layers upon layers interspersed with other layers, offering protection, offering means of absorbing nutrients, which we don't do, but for some of these flukes, that's how they get their food. That's how they get some of their nutrients. Now, unlike us, where it's an epidermis with an underlying epidermal layer, and below that, epidermal underlying dermis, followed by subcutaneous fat, muscular, blah, blah, blah. What we're going to see here with the trematodes is that the epidermal layer actually occurs in two places. You have the outer exposed to the environment, but the epidermis is also beneath certain tissue levels. And it's interesting in how you see that. The outer and the inner are actually attached to each other. They are not just so much, let me rephrase that, they're not so much attached as they are continuous with each other. They are actually the same layer, the same cell. They refer to this as a distal cytoplasm. The cell is literally in two places. There's an underlying layer, a channel, and the outer. The distal, they refer to as the cyton, and then the outer part that's exposed to the environment, they refer to this as the syncytial portion. Now let's take that and let's unpack that for a bit. A syncytia is where multiple cells fuse together. Multiple cells fuse together, but they still maintain individual nuclei. The nuclei control individual regions of this new macro cell, for lack of a better term. So that means the outer that's exposed to is all one big cell that you see coating the organism, maybe broken up into different regions. There's this channel and then this underlying portion that we refer to as the cyton. When you look here at this figure, yes, I know it's rotated to be on its side so it can fit. But what you see here is this region here, circled. This is the epidermal tissue that is exposed to the environment. This region you see here, circled. These are the underlying areas referred to as the cytons. The arrow is pointed to those channels known as the internuncial channels that allow the cyton to interact with its portion of the cell that is removed from it. And you look here, the tissue layers that are interspersed, separating the cyton and the syncytial portion. Muscle. Yes, there's going to be muscle tissues, different multiple muscle layers. There's going to be other layers of tissues that separate the cyton from the syncytial. Now it's interesting when you also look at this, if you look in the area that's again circled, you will see that there are no plasma membranes. There's a membrane along the outer portion exposed to the environment, and there's a basal membrane where the cutaneous, subcutaneous layers that that have the muscles in them come into contact with the syncytia. But again, it's all one cell. You also notice that there was what's called a spine. Okay, turns out that the tegument is also going to have some form of ornamentation associated with it. The ornamentation is going to be accessory appendages, things that are there to aid in the work of the fluke. The spines are going to act as holdfasts. Okay, the fluke is going to wedge its way up into some crevice within an organ. Say again, the liver fluke. It's going to worm its way, swiggle its way up through the bile ducts, and it's going to wedge its way there. The spines are going to be pushing pointed posterior so that the fluke cannot be pulled back because the spine will catch on the bile duct wall. So this way, even if its suckers can't hold it in place, well, maybe the spines will counteract. And if you look here, pictures, our friend schistosoma, which we spent a lot of time talking about in chapter, 3 and 4? The immunology chapters and whatnot. You see here that there's quite a few spines. These spines are going to be arranged in such a way to aid in not only its movement, but in its ability to hold fast, to hold in place. And you can look at here a different magnification on a different schistosoma. And it's going to be all around the fluke. They're going to have different roles depending upon where they're at, and they're going to be in different configurations. We also see in the tegument that there's going to be vesicular inclusions, vesicles that are going to be moving not only within the cell, intravesicular, but from tissue to tissue, taking from one cell to another cell, either within the same tissue or in different tissues. Turns out the plasma membrane of the epidermal cells we're talking about here, the syncytia portion, it's constantly being damaged, constantly needs to be turned over. So these vesicles are being made, they're being transported through tissue layers, and they will be added to the syncytia basal membrane and then transferred through the cell to the plasma membrane, or the cellular membrane that's exposed to the environment. Turns out many of these flukes are constantly shedding, kind of like how we're constantly shedding. They're also constantly shedding epidermal tissues, epidermal layers, portions of the membrane, especially when they're going to be opsonized with antibodies. Instead of, having to deal with all those antibodies studying, they'll just shed it off. And it's replaced from underlying tissues, and it just keeps going and going this way. Some of these vesicles can be used by other flukes to deliver different proteases, different enzymes. These will be Golgi-derived vesicles, so they're going to start in the cyton, be transported through the intranucleal processes, the intranucleal channels to the syncytia, then to the syncytia membrane, and then out of the cell. Now, it's important to notice, as you see here in this figure, from the myristidium stage of fasciola hepatica, that it's not just the adult that has these very advanced tissue layers of the tegument. You look here, as again, this is the myristidium. This is going to be a larval stage. Multiple layers, multiple tissues, muscular, epithelial. Underlying germ cells are going to be making and replacing damaged epidermal cells. Those damaged epidermal cells will just be shed and cast off. The ciliated appearance you're going to see here with many of the epithelium, specifically with the myristidium, it's not the 9+2 cilia that we've seen with the protist. Here, it's going to be one cell, multiple cilia, almost to the point of being like villi, okay? I've seen it discussed both ways. You can have the 9+2 microtubules, that we've seen before and talked with flagella and cilia in, eukaryotic single-cell things. But it can also be more closely related to the microvilli we see in our endothelium lining our gastrointestinal tract. The microfilaments are acting as the cytoskeletal structure to support the microvilli here. So I've seen it both ways, so I'm not going to spend a lot of time fixated on this. But when you look at it, notice again, you're going to see the beginnings of the syncytia forming here. There are ridges, there are areas where the syncytial cells are separate. So it's not one continuous cell wrapped around, acting as the epidermal layer. It's going to be multiple, but they are multinucleated, large cells. There's going to be what they refer to as ridges in between, giving some separation. Underneath those, you're going to find the many accessory cells, many accessory tissues, basal membrane. Below that, you're going to see the muscle layer. You're going to see different layers of tissues, different layers of cells and organs. And when you look at the development, here you see the formation of the tegument for the cercaria. Okay, the cercaria development means it hasn't still going to be in the rhodea, and the rhodea and the right into the juvenile stages where it's been released out of the snail host. You'll see that the tissues, the architecture, completely rearranges. Okay, certain tissue layers will actually increase in volume, increase in, just become a higher order tissue, developing towards an organ system. Other tissues are going to lose complexity, and they're going to be absorbed, become part of different. Those cells are going to be either broken down or recycled to become something different.